Okay, so today we're gonna finish the antibiotics. So we get started. So finish up on Tuesday, last week Thursday, we talk about what is antibiotics. And I said it is metabolic ingredients coming from a microorganism to kill or inhibit another microorganism. And most of the time, the antibiotics coming from a fungi. So for example, the penicillium created penicillin. Then streptomyces created tetracycline. And most of the antibiotics at the very beginning, they are natural. Except for sulfalaminates, which is a sulfur drug. And sulfur drug, it is manufactured. But we still call them antibiotics. But right now, these days, all the antibiotics we are using, they are basically half and half. Half the ingredients from the nature, they are cultivated, but they are manufactured your biochemical or biotechnology. So they are not like growing penicillin, they generate the penicillin, then you go ahead to do the treatment. It's different. So we talk about what is the chemical therapeutic agents, what is antibiotics, and what is antimicrobials. Then we talk a little bit about the history, and you can see in the history we try to solve the problem like syphilis, sexual transmitted disease, and I said it's very tough. That's the oldest sexual transmitted disease pathogen in the world, which is a sarin, which is a parasite. So it started from 1912. Then everybody heard about is Fleming's discovered the first antibiotics, which is penicillin. And I also talked earlier, tuberculosis is really tough. So in 1944, Bucci and Wasman discovered streptomycin used against the tuberculosis. And after 1950, so the recent 70 years, we use a lot for the antibiotics, but we also generate a problem, which is a universal global risk with antibiotic resistance, which means the pathogen become less useful if you treat them with antibiotics, more and more resistant. Some of them become a multi-drug resistant. This is an example of a penicillin. So you have a penicillin, which is a fungi growing there. And then if you do the streak, see those greenish area, which means bacteria is growing, except surrounded by the penicillin, they are not. They are transparent zone. That means which is inhibited with penicillin when it gets close. And we're going to talk about that later. This is one of the ways to determine the antimicrobial effects of antibiotics. We had several things I want to mention here about those terminologies. Then we go to talk about how we tested them. First of all, we want to say antibiotics, when they talk about a bacteria, let's say penicillin can kill E. coli. We say a bacteria is susceptible to antibiotics. But for human being, we cannot just randomly taking antibiotics or give a shot of antibiotics. Lots of the time, some people will have allergic reaction. So for human beings, if they are allergic to antibiotics, we do not call it susceptible, we call it sensitive. So sensitive and somehow, which means allergic. So in the old time when I was young, like 4 or 5 years old, if we go to see a doctor, let's say I want to give a shot, they want to give me a shot of penicillin, they need to do a skin test. If after the skin test, about 72 hours, the skin shows swelling, which means you're susceptible, you're sensitive to penicillin, you cannot use it. And I will say, when I was young, 40 years ago, the penicillin we can only take in is by shots. There's no methods by the time to do the oral administration, but right now we can just take in the pill orally. There's a development history about that. So here are some of the concepts. Selectivity toxicity, ability of drug to kill or inhibit the pathogen will damage host as little as possible. That's called the side effect has to be little as possible. The second concept called the therapeutic dose, which is drug level required for clinical treatments. Usually for antibiotics, we want them to be microgram. So on the disc, when you see P10, which means that is penicillin, the dose is 10 microgram. Obviously, the smaller the better. We want them kill bacteria, but also we don't want a human being has side effects. Toxic dose, we just said, drug level at which drug becomes too toxic for patient. Now, therapeutic index, that is an index, which means you have a toxic dose divided therapeutic dose. What do you think this dose should be? The index should be the smaller the better, because we want this the smaller the better. We wanted the larger the better. We wanted the toxic dose is the larger the better. We want a therapeutic dose, the smaller the better. So based on the mass, we want this index, the larger the better. Think about it, if toxic dose is the same, therapeutic dose we want to be smaller. Instead of using 100 microgram, we wish 10 gram could be successful. Toxic dose, we want them the higher the better. Because even if we use 200 microgram, you still don't have any side effect. So if you do the math, the index should be the higher the better. Side effects, we already said, is undesirable effects of drugs on host cells. So next two are the same thing. Narrow spectrum drugs and broad spectrum drugs. Narrow spectrum drugs is only attack a few different pathogens. Broad spectrum drugs is attack everything. Basically, narrow spectrum, which means only gram positive or gram negative. Broad spectrum means both gram positive and gram negative. Cyto agent, the terminology called cyto, which means it kills microbes. Static, which means it inhibits growth of the microorganism. So these are some of the general characteristics, or we say it's a concept. Now, how we gonna determine antibiotics? This is a very interesting topic. So think about this way. In the very old time, in the very beginning, you go to see a doctor. You are coughing and sneezing. You have a streptococcus pneumonia. Let's say all streptococcus pyogenes. We said how you have to use antibiotics immediately. But we need to determine which antibiotics you should use. And we also going to determine what is the dose you should be using. So therefore, we have a lot of the tests Now basically we have three tests we can use. The first test is called the Kirby-Boa test. So how we do the Kirby-Boa test? So first of all, I had a bacteria there, okay? A bacteria, we're gonna grow. The bacteria isolated from the patient's sputum sample, let's say. Let's say we get a Streptococcus pneumonia. Okay, I'm gonna be to grow it. And then I wanted to use them to do the test. But the first thing first, how much bacteria we should be using? There is a standard called 0.5 McFarland standard, which means roughly 10 to the power of 8 cells per ml. Now how we do this? Usually the bacteria growing, let's say grow at TSB chart, soil broth, we will dilute it 10 times in 0.1% salt solution or saline solution. Okay, let's just say saline solution. Then we got to compare to a standard sample, usually made by silicone. The turbidity looks like a bacterial growth, but it's not a bacteria. Then you compare these two tubes. This is a tube right here. If the turbidity looks similar, we will say it is approximately 10 to the 8 cells on the tube. Then you find a bacteria agar. This bacteria agar is not just a general agar. It's a very special agar called Muller-Hilton agar. A special agar to do antibiotic resistant test. So how we do? We're using cotton swab to take the bacteria from here. It's a cotton swab. We took it goes here. We do very gentle, averagely covered on the surface. Put the bacteria there. Then the cotton swab to 90 degrees, we do a second swab. Usually we did this in the lab. We do that averagely. Then what we do, we need a disc dispenser. The disc is usually looks like that. They have a lot of small pieces. Each piece is a circle. This is called disc. They containing somehow like P10, which means penicillin 10 microgram. Sometimes you will see PE 20, tetracycline 20 microgram. The disc containing antibiotics. So how we do? We put the disc, goes there, we press the button, it will be dispenser. Okay, so what it looks like. The disc is covered this area. That is P, let's say 10. That is tetracycline 20, let's say. Then this plate incubate 35 degrees Celsius 24 hours. let the bacteria grow. Most often the time, we will see inhibitory zone surrounding the disc. We also see an inhibitory zone surrounding the disc. So it looks like this. What this means, the bacteria is inhibited. We see the transparent zone. Then we're going to use a ruler to measure the diameter. And we're going to compare the table. So, let's give an example. In the table, which is standard, coming from American Antibiotic Association, they have standards for every single antibiotics. So for penicillin, they usually set up S means susceptible, I means intermediate, R means resistant. Usually the number is 18, 14, and 10. For tetracycline, that's 20, 15, and 12. The unit is millimeter. So let's say we had a streptococcus pneumonia here from this patient. And then we put a disc there after incubation. We measure, this is about, let's say, 9 millimeter. If we get 9 millimeter, what this means? Which means the streptococcus pneumonia from this patient, they are resistant to penicillin. So penicillin cannot be used for treatment of this patient. So what are we going to do? Yeah, let's see tetracycline, for example. Tetracycline here, we see the inhibitory zone is 22 millimeter. Okay, good. That is larger than the number here. So, we will say this streptococcus pneumonia from this patient is susceptible to tetracycline. So we can use tetracycline to do the treatment instead of using penicillin. But does that mean this dose is good? Probably not. Or we have to do a test. The test is further called minimal inhibitory concentration test. So how are we going to do it? Let's say we're using Kirby-Boyle test. We already find this patient is good at using tetracycline. Now we're going to do the test. So how we do the test? Called MIC determination. Because we want to be the concentration is smaller the better. We don't want to be anything huge. The reason is maybe the patient use 20 microgram of tetracycline will cause a very severe side effects. You know that tetracycline could cause dishearing, yellow teeth, could have all kinds of those side effects. We'll talk about the mechanism later, okay? So how we do it? Okay, we grow a bacteria, Streptococcus pneumonia. Okay, then we prepare lots of the tubes. Lots of the tube. Let's say we started the tetracycline. Yes, 64 microgram per ml. So in here, we are going to add in 64 microgram per ml into 0.1%, saline solution. Then most of the time we are doing a serial dilution. And this serial dilution is, we call it a twofold. Every time divided by 2. So the second one is 32, then 16, then 8, then 4, then 2, and then 1. Okay, we have a saline solution. Most of the time, we containing trophic soluble loss here, because we want the bacteria to grow. So instead of using saline solution, we also dilute it in TSP. Okay? And then we inoculate the bacteria inside. We inoculate the bacteria. And then we incubate it at 35 degrees Celsius for 24 hours. At the same time, we need to have the control. Usually the TSP solution with Streptococcus pneumoniae, we call this the positive control. And we also need another control, which is 0.1% saline solution. We also gonna incubate these 35 degrees Celsius 24 hours. Okay, then the second day, we see the results. We wanted to see there is no bacterial growth here, which means the saline solution is sterilized. If the saline solution, you see the bacterial growth becomes turbidity, that's not good, which means cross-contamination. TSP with pneumonia, we want to see its turbidity, which means the Streptococcus pneumonia is good. If this TSP doesn't grow, which means the bacteria may not be survived already. So that's called a positive control. This is usually we call negative control. Now we look at these testing tubes. First one microgram, we see turbidity, 3+. Second one, we still see turbidity, but less, 2+. This we still see turbidity, but more light, 1+. Start from here, we see transparent. The first one, showing transparent tube, is inhibitory concentration. We call it a minimal inhibitory concentration. In the older time, this test has to be done individual, corresponding to each pathogen. Let's say 8 microgram is effective for tetracycline for Streptococcus pneumoniae in a boy named John, let's say. If a girl comes named Jessica, she has to do another individual test, because the Streptococcus on Jessica does not mean it's the same as the Streptococcus from John. So that's something we have to do, it's independent. Now right now we all have those rapid testing kits. We can get a determination very quick. But in the older time, and the principles are also the same, is you have to use tubes to determine the minimal inhibitory concentration. And the good thing is we will clearly to know what is the MIC, because we want to use it as little as possible. So these type of tests, we call it quantitative test. And these type of tests, qualitative test. Now we have something between qualitative and the quantitative. We call it a semi-quantitative test. It's called the E-test. So E-test, how we do it, it's a little bit different. We grow the bacteria. We're going to put a strix there. This guy is a disc or a strip. It's say a medical paper containing medicine, containing antibiotics. The dose, which is usually also unfolded, two-folded. You put a strip there, you inoculate the bacteria. You want to see a first dose in the intersection of transparent zone and the bacteria community. That is a number for MIC. So right in here, you can see these are the number we are looking for. That's the number we are looking for. Why we call it E-test. It is a quantitative test. Okay? Okay, we want to talk about antibiotic, antimicrobial mechanism. Here we listed, there are basically 4 different types of the targets for antibacterial mechanism, which is inhibit cell wall synthesis, inhibit protein synthesis, inhibition nucleic acid synthesis, inhibition metabolic pathways, and inhibition cell membrane integrity. So the first question, which mechanism among those 5 is most effective for bacteria and also less risk for human being? Which one? The answer is very easy, is on the top, to inhibition of cell wall. But you need to know why. Because bacteria has cell wall as a prokaryotes. Human being, an animal, and lots of the plant cells, they do not have cell wall. What this tells you, if your target for antimicrobials or antibiotics is for bacterial cell wall, then our human being don't have cell wall, which means you don't have a target. So the side effects will be much less. Okay, so that's something we want to talk. Cell wall. We talked about penicillin. We talk about lots of the other antibiotics. This is a picture really amazing. We talk about the principles. These are the 3 basic cell wall structures will be inhibited by 3 very typical antibiotics. First of all, we want to talk about is penicillin. Penicillin, they have a very special structure called beta-lactam ring. This is a unique structure for penicillin to killing bacteria. Why they can kill bacteria? You can read it. They'll interfere with the formation of the peptide side chain between adjacent strands of the peptidoglycan by inhibit penicillin binding protein. Here is some basic knowledge we need to know. I usually don't teach this in this class, I teach in the other class. Bacterial cell wall has two major structures. Is modified sugar and amino acids. What are the two modified sugar? Is NAM and NAG. The full name is N-acetylmuramic acid and N-acetylglucosamine. We just skip it, just call the acronym. They connected like this. And you have another chain. Do the alternate arrangements. Okay. Now, how they connected with each other? With amino acids. I just use AA. They connected. Amino acid, you know. Amine group, chiral C, R, and the carboxy group. Is that right? You learned that. Based on the side chain of the R, we have 20 amino acid. If R is H, that's glycine. CH3, that's alanine. Then we have like phenylalanine, histidine. Like in the amino acid in the milk is lysine. That's amino acid. So they're connected with. Okay. So where's the beta-lactam ring gonna come? Side chain of the adjacent strand. So they are attacking here. Okay, they are attacking here. Now the question is, if the patient is using beta-lactam ring based antibiotics, let's say penicillin, get resistant, what we should use? Then go ahead to see vancomycin. Vancomycin binding to the amino acid chain of NAM molecules interfering with peptidoglycan synthesis. Vancomycin target here. We mentioned about vancomycin earlier. I don't know whether you noticed. When we mentioned this, Clostridium difficile. What is cause Clostridium difficile? We are using antibiotics too much, intensively, for a week. So a good example will be an older lady in the hospital using antibiotics, penicillin, about a week. Have pseudomembrane colitis and water diarrhea. Then one of the methods to do that, and I said it's very ironic, because the problem caused by using antibiotics, but we have to use another antibiotics. That is vancomycin. Why we can use it? Because the target of vancomycin is different from penicillin. Penicillin is attacking amino acids. This is attacking NAM. That's why we can use it. So that's vancomycin. Okay, let's say penicillin and the vancomycin both doesn't work. What we have to do? We have another choice, using bacitracin. Because let's say they are fighting. Okay, inside, this guy doesn't work, this doesn't work. I will do, let's say when they build up something, bacteria need nutrition. I'm blocked here. I don't let you come in. That's what bacitracin does. But you always need to know, if you use bacitracin, you prevent their coming. The bacteria are smart, they can generate something by themselves. That's why lots of mutation will be generated. So, basic tracing basically is inhibit bacterial growth because it blocks the nutrition coming. They are not really killing bacteria. So basic tracing impacts basically static instead of cyto. We heard about the basic tracing already where? A disc. The components of A disc to differentiate the Streptococcus pyogenes and Streptococcus agalactiae is basic tracing. Okay, so this slide is actually very interesting. Tells you three very basic and already been well-known antibiotics, how they inhibit the bacterial cell wall. Now I want to go back to talk about this penicillin a little bit. Penicillin is interesting. There are lots of things we could talk. There are different generations of penicillin. In the very old time, about 70 years ago, when they have penicillin, we call it penicillin V. Penicillin V only can do is muscle injection. Give a shot, which means where they give in the butt, give a shot. And usually the human butt will be swelling, become red. So sometimes they can make a joke for the kids if they are naughty, we'll give you shots of penicillin V because it hurts a lot. The reason is antibiotics is large molecular. It's in the muscle, it's migration, which means it goes everywhere, it's hurt. But they can only do muscle injection. Why? Because our stomach acid will hydrolyze it. They are not resistant to stomach acid. Stomach acid is very low pH, very acid, 1 to 2. So people find this is not very good. Okay, they developed the next generation, penicillin G. Penicillin G started to have oral pills. They are acid resistant. But same as penicillin V and penicillin G and penicillin V are narrow spectrum. What this means? Only for gram-positive bacteria. Not very effective, or we will say minimal impact for gram-negative bacteria. That's not good. Therefore, people generate a next generation. This is everybody knows, methicillin. People are using a lot. But we get a problem. What is that? MRSA, methicillin-resistant Staphylococcus aureus. We using a lot. Then people say, okay, we did a lot for methicillin. We need to be smart, so they generate next generation, amoxicillin. That is why today you go to CVS, you physician, if they give you antibiotics, it's all amoxicillin. Or it take started from later stage of methicillin and amoxicillin, they are basically manufactured. They are not natural products. Penicillin V and G, they are natural products. This is 75% is bio-manufactured. However, amoxicillin used way too much. They become amoxicillin resistant. They become multi-drug resistant. So that's why today you have the feeling, whenever you use antibiotics, it took a while to recover. It's not like you give a shot, can be fully recover tomorrow. So several things I want to also add it up here. Antibiotics, although it is a breakthrough in the history, but I want to tell you, most of the time, antibiotics is more effective for gram-positive bacteria than gram-negative bacteria. Why? I think you can think about this. Is when we talk about gram stain, what is the major difference between gram-positive and the gram-negative? Is this heavy chunk here. Peptidoglycan. Gram-positive bacteria very sick. Gram-negative bacteria very sick. If you have a sick peptidoglycan, antibiotics has a more obvious target they can work on. This is minimal. Second, antibiotics most often the time is more effective for growing bacteria, which means the bacteria is growing, they are building. It's easy to turn it away. Think about you are building those blocks, briquettes, building a house. When you're building, you need to turn it off or damage it. If you already built a house, you tear this one off, much more difficult. So, there is a condition for that. Gram-negative bacteria, to be honest, not very effective. Otherwise, you will not hear any of the recalls or outbreaks about E. coli, O157H7, and Salmonella. Those are all gram-negative bacteria. If you eat undercooked beef steaks, you only need to go to see a doctor, give a shot of penicillin, they solve the problem. Obviously not. Because those gram-negative bacteria are difficult, more difficult to cure it. Okay? So that's something we want to talk about. Okay, let's say a patient using all those three doesn't work. What we do? You have to use something different. We have to do something inhibit protein synthesis. So regarding protein synthesis, we need to talk about the structure. Which cell machinery is for protein synthesis? Ribosome, is that right? Ribosome have two units, 50S and 30S. Now remember inside here, 16S rRNA, then here, 23S rRNA. Then later on this becomes a good one, so we say 16S rRNA biotechnology. That's where they come from. Okay, add up together is equals what? 70S, not 80S. Why? Because that's Vandenberg units. It's testing precipitation, not for molecular weight. Antibiotics can do target protein synthesis, which means either 30S subunits of the ribosome or 50S subunits of the ribosome, because protein biosynthesis is in here. But what a bad thing. Our human being has protein. So when they are attacking here, they are attacking a bacterial protein. In other words, more or less going to attack human protein. That's why all these antibiotics listed here, more or less, has side effects. Some of them are very severe. For example, chloramphenicol. Chloramphenicol is the first generation of broad spectrum antibiotics. It's almost cured everything. But right now we don't use it unless life-threatening situation. Why? they will decrease the function of bone marrow, which means if some people using chlorophenol too much, you will have red blood cell decreased. The bone marrow is making the blood. You have the blood cells decreased dramatically. That's not good. Immune system will be decreased. Tetracycline is not good. Tetracycline is no longer used, only for animals. Even right now, the veterinary science area does not use that too much. One reason is it causes antibiotics too resistant to way too much. Second reason will cause yellow teeth and hearing loss. All others, streptogramine, aminoglycides, merolides, they all have a little bit side effects. Okay, that's something different. People say, how about the nucleic acid synthesis? When every time you see nucleic acid synthesis, you will see, well, that's very good. Because it will prevent the DNA replication. But I tell you, it's least effective. Why? What we learned in your different classes about the DNA replication is a standard model. In the real life, bacteria have all kinds of ways to combat if there is a harmful attacking for them. They always have a bypass for nucleic acid synthesis, and lots of them we don't even know. So, if antibiotics, if they are attacking nucleic acid synthesis, it's least effective. But why we need those? This is what we call laboratory tool. So, give you an example. I have a beef. Okay, that's a beef. I want to do a research. I want to see 5% lactic acid, whether they can kill E. coli, O157H7. So in the lab, the first thing what we do, we we add E. coli O157H7 there. But then the question is, how do you know this is the E. coli O157H7 you added to differentiate from the beef already existing O157H7? We give them a marker. We usually give them rifampicin resistant E. coli. How you do it? In the agar plates, you add E. coli O157H7. Then the plates is already containing 200 parts per million rifampicin. After incubation, 35-24 hours, you will see some colony growing there. You see some colony growing there. Then this continue to do 5 generation subculture. That E. coli will become resistant E. coli O157H7. Can be used in the research. Because we know the E. coli O157H7 we added is not from the beef itself. Is what we artificially inoculated. But here is something. These type of the resistant is only working for chromosome, is not attacking or work for plasmid. So every time when you cultivation of E. coli, you need a rifampicin there as an inducer to generate resistant bacteria. So inhibition of nucleic acid synthesis is only for making the bacteria used for the research, not really for medical reason. This you already know in metabolic pathway for sulfur drug. So you know the story. This is called PABA. PABA is a precursor for folic acids. Is that right? So people say folic acid is very useful for pregnant women because it will protect the baby do not have neurological symptoms. PABA, para-aminobenzoic acid, is a precursor for generate folic acids. And the bacteria must to add PABA in their media for them to grow. Now we have a sulfur drug. Looks like the structure is very the same. Now you know these things when you have a substrate. This is the enzyme, you go there. It's like a key and a lock. They will be connected with each other to have biochemistry reaction. You have this is PABA, and you have the same as a sulfur drug. If the other structure are the same, they could competitor or compete with each other for the site of the reaction, of the enzymatic reaction. So instead of the PABA there, the sulfur drug could be there. Therefore, the folic acids will be inhibited. So that's what's the story for that. So metabolic pathway is one of the methods to inhibit bacterial growth. Because bacteria always find an alternative way to generate those metabolic ingredients if they needed it to survive. The last one, polymixing. This usually causes leakage of the cellular contents. Change membrane permeability. When you see these slides, you probably doesn't recognize what type of antibiotics it is. But if I tell you, you absolutely know. This is ornament from CVS. Where we use it? If you have a skin itching, it's a countertop antibiotics. Only used on the skin surface because it's inhibition of cell membrane integrity. This is least effective because the leakage of cellular content used on the skin surface. Ornament is a good example. A very good slides. Give you a conclusion of antibacterial targets. You should keep this Mark 3 star. In the exam 2, we'll have one question, talk about antibacterial targets. Let you do the multiple choice. Okay? So the material what we're going to be talking today is done. But I want to give you, have a little bit of minutes to talk about the exam 2. You guys absolutely need to print it out. The after class reading material here, and also read it. At least go through it. These will be very useful for prepare exam 2. Especially the case study. How to do a case study? Why we have this? Let's give you example, okay? A 2-year-old girl is admitted to hospital with massive issue destruction along her right arm. The skin is a violet color and large fluid-filled blisters are present. The patient has a fever, a rapid heart rate, low blood pressure, seems confused. Her mother informed the physician that the children had been recovered from chicken pox. And the past 2 days had frequently been scratching at chickenpox lesions on that area of her arm. Then they do, once the area appear to have become infected, the infection spread very rapidly. A gram stain exudate from the infection disease tissue find a gram-positive cocktail in chain. So which pathogen? Gram-positive cocktail in chain, which means it's Streptococcus. So you're gonna do look at here. This is gone. This is gone. CDE. Streptococcus agalactia, pneumonia, and pyogenes. Okay. This relates to the chickenpox. So you go back to read. So let's go here to Did you see it? Anyone can become infected with group A strep. Breaks in the skin like cuts, surgical wounds, or chickenpox blisters provide an opportunity for bacteria to enter the body. So which one? Where we talk about here? What is group A strep? Streptococcus pyogenes. So the answer is Streptococcus pyogenes. That's how you're going to do the exam question. Let's look at the second one. 80-year-old female from nursing home to the hospital because she's suspected for having pneumonia. Okay, that's good. Pneumonia. We talked all the time. Chest pain, chills, fever, shortness of breath, productive cough. Cough, chest pain, and fever. So that's what you should do. Chest pain, fever, and cough. A gram stain of the sputum reveals numerous white blood cells and the gram-positive diprococcal. What is that? Which bacteria is gram-positive diprococcal? We talk all the time. Streptococcus, pneumonia. Yes, that's how we do it. I mean, this one, if you studied hard, you studied really interested about this, it's very interesting. I mean, major students have to remember. It's a closed book. Oh, they're confusing all the time. Oh, because they have multiple choice. But you can read it. You can open the material to read it. But you better read it when you have a little bit of time before the exam. Okay, otherwise you're going to spend a huge time to find it. But the question will be like that. Okay, so let's give you an example. And you will have also after class reading too, for example. And I'll give you guys a practice exam question this time, okay? A short version, then you can practice. I'll give you the key. We have a case study right here. Right there. What do you see? Temperature was normal. It has not never received the vaccine. Right here. Vomiting, difficult swallowing, difficult breath. Membrane exit covering those tonsils. What's that? Colobacteria, diphtheria. Right on the top. Yes, diphtheria. And then we talk about the story, and then we have a case study. They're easy. So, what are we going to looking for? Are we going to read all these? Highlight the characteristics corresponding to each bacteria. So that's some work you should do. Okay, highlight it. Then it's easier to find it. So that's the exam questions. Caleb said we're going to have the same thing. Hemophilus influenza, another big one. Okay, so we will do is based on the calendar. 26, Nettie Fresher will do a guest lecture. March 3rd, I will do a review because it's so many bacteria. We're going to talk about, spending 60 minutes, talk about the review, then you do exam on March 5th. Okay, then you go ahead to have a, we can teach a little bit here, then we got to go ahead and have a good spring break. Okay, so that will be the plan, how we're going to do the second exam.